So I don't know what it's like for you when you have an epiphany, that moment of realization that about something that your brain has been dwelling on for a while. With me, when I have an epiphany, which is rare, I end up with this feeling of elation, very closely followed by this feeling, geez, I should have figured that one out a long time ago, because it was sitting in front of me for days or weeks or months or whatever. So my particular epiphany, which is going to be the topic of my talk today, is the value of our artist brothers and sisters and why they do what they do and why it is important for some of the things that I'm doing now in my dual worlds of seeing kids with genetic problems and doing the genomic testing that will allow us to understand what's going on with them. So I'm hoping that you're not all as poorly informed as I am about the world of art, and some of this will be completely obvious to you, in which case my hat is off to you because I'm just not up to it. And I thank Monica for all of the work that she's done to create these very handsome-looking interpretations of our faces. Very good-looking audience today, by the way. Congratulations to all of you for that. So we live in the world of big data, which is, MRI images, genomic images, the life of a cell, the sort of immunohistochemistry you see in a pathology report. We could sequence all of your genomes in a few weeks if I sent it to a couple of genome centers in the United States. We'd have the data back, but we'd be pushing hundreds of gigabytes or terabytes of information by the time we got that information back. It's very difficult for anybody in the first row to take those genomic data and do anything with it. So that's kind of a problem. And it's what I call the Barbara McClintock problem. So Barbara McClintock is the woman on the right here. She's the woman on the right here. She was a woman in science. She was a plant geneticist studying corn. Ronan thought those were teeth. Those are ears of corn. She was one of the few women who studied science during this era. It was an era when, to look like a serious professor, you had to smoke a pipe. I've yet to achieve that exalted status. But what Barbara McClintock did was she was such a good scientist, such a good observer of the patterns of pigmentation on these ears of corn, which were genetically driven, that she was able to infer that what was happening was that genes were jumping around the genome. And this was in an era where we're just trying to get our heads around the idea that DNA existed, that it encoded genes, and that genes in some way had physical relationships to each other. And she was saying, yes, fine, all of that is true, but they can also jump around the genome. A very challenging concept. She was right. She got the Nobel Prize in Physiology and Medicine in 1983. And she was asked by her biographer, why is it that you managed to figure all of these things out? A very nice biography of a scientist or of a woman in science by Evelyn Fox Keller. And Evelyn Fox Keller said, tell us why it was that you made these discoveries as opposed to other people. And very unusually in this book, Barbara McClintock's answer almost took on a mystical tone. She was talking about how you have to be open to what the organism is trying to tell you, what the thing that you're studying is trying to communicate to you. Be receptive to those messages and have a feeling for the organism. Now, in her case, she was talking about ears of corn. In the case of medicine, we would talk about a gestalt, some sort of gut feeling about there's something about this kid that really worries me, or there's something about this presentation of pneumonia that is not quite what I've seen before. And I think we all have a sense of what she was trying to communicate. But the problem today is, Barbara McClintock was studying ears of corn. Today's Barbara McClintock, who could be here, she could be in my home institution in New York, she could be anywhere, we have now abstracted that information into the digital domain. So have we prevented Barbara McClintock of today from making discoveries and having this intuition? So I started to think about this problem and think about how other people have addressed this in the past. And it's not kind of known, even for New Yorkers, and he was our mayor, how Michael Bloomberg made his money. He was fired from an investment company called Salomon Brothers. He was given an amount of money. And he decided that what he would do is tackle an issue that was prevalent in finance at the time, which is currently exactly the same as we have in genomics. If I wanted to understand in that era, as a trader, whether I should buy or sell, make a trading decision, I would have to go to a colleague of mine who would access the information which was out there on the World Wide Web, and that person would give me some sort of representation of the data, and then I could make a decision. But I was not able to access the data myself. And what Bloomberg decided to do was to create a very simple keyboard so that these guys would be able to navigate very simplistically and to get to these visual representations of the data. And this is exactly the sort of thing that he had in 1983. And while the update of the system in 1990 was looking more slick, and the current version of the system is very polished looking altogether, it still retains those basic design principles of the almost toy-like keyboard and a lot of visual representations of data so that it allows you to make decisions that in the financial world will be more informed. Our colleagues in astrophysics, they are very collegial, and they figured out that what they could do with all of the vast amounts of data that they were generating is share them on the World Wide Web. And you could get on your phone or your tablet right now and look up Virtual Observatory and do this. Because what they were realizing was that people were pointing their telescopes at the sky, getting light images, X-ray images, infrared images of the same areas of the sky, and you could overlay these and see how they're relating to each other. In this case, looking at the Crab Nebula. So another visualization technique. And one of my wife's colleagues, Jim Rawson from Georgia, gave me this. He says, if radiologists were trained in a different way, they'd be able to look at this and make a diagnosis. They'd say something is happening at about row 290 and about column 304, where there are these increased values in this tabular representation of data. But while a lot of imaging today is digital, the choice that has been made about how to represent these data back to us as physicians is to create a visual representation. In this case, this is exactly the same data that are being shown. And now what you can see at those particular rows and columns is that the increased signal is at the point where the ureter enters the bladder, and it is obviously a ureteric calculus. So in each of these cases, what has happened is somebody has figured out how to format the data in a standardized way, which is a technical issue, but more importantly, they figured out how to take those data and turn it into a representation that's visual and useful. Okay. So I started to think, if Bloomberg figured this out, if some astrophysicists and other people have been making these sort of decisions, how has this been done historically? These are relatively recent examples. So I'm going to get to the four-faced Buddha in a minute. And the first thing that I looked at was the heat map. The heat map is a very common representation that we use in the world of genomics to try to understand lots of things simultaneously in two-dimensional space. So what you're seeing here in the columns are patients, and what is being depicted in the rows are epigenetic marks in the genome, which you don't need to know anything about, except to point out that the people with the disease are shown in black, and there's a cluster of them here on the right, where the patterns appear to be somewhat different to the people who either have a prodromal form or who are controls. So it allows us to see a lot of different places in the genome and a lot of people simultaneously. I thought, that's very clever. Who came up with that and when? So I started looking at the history of data visualization. And if you go back to the Paleolithic era, the era of the diet, what they were doing in modern-day south of France was they were drawing pictures of beasts on the wall. And if you look at the dots, they have actually been represented, they appear to represent things like Orion's belt and Pleiades constellations in the sky, which is not that challenging. You're basically saying, I see these dots, I'm going to make the same dots on the wall. But in the modern-day Czech Republic, on the banks of the Dia River, something much more interesting was happening. So I only want you to notice here that the Dia River is quite sinuous, and it's forming these little sort of cutoffs, these oxbow lakes, or what they'd call in Australia, billabongs. So if you notice that sinuous nature of the course of the Dia River, one of the things that they did in this region was they did an archaeological dig, and they found a mammoth tusk. And the mammoth tusk had these engravings in it, which are, you can see depicted by this line diagram here. And what they recognized these were showing was the course of the Dia River and the terrain in the intervening areas. So in this Paleolithic era, somebody who's probably about 5 feet tall was able to imagine what it was like to be about 1,000 meters in the sky, looking down, and depicted that, which suggests that we innately, as a species, have this ability to think in the abstract terms that allow us to create data representations and have the ability to recognize it as valuable. You have to go forward to about the 14th century before we started to take what would be a tabular representation of data like this. And you look at this and you can see a kind of a relationship, but you have to kind of think through it with your head. Nicole Oresme, 100 years before Gutenberg came up with the printing press, represented these sort of data with what would be the precursor of the modern bar graph. And you think about it, this is the first thing that you'd reach for in Excel if you wanted to represent data in some way. It was 1300s. They wanted to see how people were measuring the longitude between Toledo and Rome. And this is basically the first representation of statistical data, looking at error in measurements. And some people were measuring it as being relatively short distance, others a longer distance. It looks like maybe there were two measuring instruments because it looks like it's bimodal representation. They were both wrong. That's the actual longitude. But this is an example of trying to show statistical data. This is a very cheerful graph. You read along the x-axis, you find your age, you go up to the curve, that's how many years you have to live. And it takes the bar graph idea and then it talks about a continuous distribution function. The idea of representing data in terms of the space, quantitative data in terms of the volume of space on a page. The idea of taking cartographic data and having quantitative data shaded in some way, which is starting to look a little bit like a heat map. I like this one. It was a British Army engineer who represented the different routes that were coming in and out of Dublin and the volume of traffic on each route in terms of the width of the line, creating what was the flow map. And this was the precursor to what has been described as the greatest data visualization in history, which was the representation of Napoleon's march on Moscow. So in beige, what you're looking at is the flow diagram of the army leaving Poland to go to Moscow, and in black, coming back from Moscow. And the width shows the size of the army. And at the bottom of the graph here, they're showing the temperatures of 0 degrees dropping to minus 30 degrees centigrade as the army is coming back. And you can see everything that you need to know about why this army was ineffective and why all of the soldiers basically died of hypothermia. If you've done any epidemiology, epidemiologists will recognize this map as being John Snow's map of an outbreak of cholera in London, where each dot was shown in terms of a case of cholera. Each pump was shown as an X, and the pump in the middle was the one that John Snow said had to be causing the cholera. He removed the handle of the pump, the cholera outbreak went away. This is the first scattergram by Francis Galton representing height versus head size. I've got a very big head. This is the Venn diagram, which is the simplest idea possible, again, 1880. And in the same era was Toussaint in France, who came up with the heat map. And so this is an interesting representation. He was basically a very enthusiastic Frenchman, a very enthusiastic Parisian, who was tasked with the job of creating a representation of all of the information about the arrondissement in Paris. So while most of this book, which you can actually see completely online in Google Books, is tabular information, towards the end he decided to create some maps like the Dupin maps that I showed earlier, where you took the quantitative information in white, sort of pink, blue, and so on, and represented it so you could see what sort of patterns there were geographically within the city. But then he thought, okay, I've created 40 maps. Is there a way I could condense it even further? So if you look at the Louvre, the first arrondissement, it's in white here. The second map shows it in pink, the third in blue, and then number 40 is in white. What he did was he created this two facing pages, white, pink, blue, and then back to white for the first arrondissement and for each of the other arrondissements in the Paris area, and created what he called modestly the Lua scaleogram, and what we would call today the heat map. So what was in common with all of these people was that all of this happened before 1900. Very few of the tools that you'll pull up on Excel to represent your data have been invented since 1900. There has actually been a pretty fallow period in the last century where we have not been doing anything innovative apart from a few exceptions that I'm excluding just so my story sounds better. But the thing that I think is a real lesson out of all of this is that the people who came up with these ways of visualizing data were not only good with numbers and good with visualization, but they were also end users of the data. They understood why the data should be represented in certain ways. And they had the ability not to have this just locked up in their heads, but to create a product, something that would get it out into the community and allow people to use it. And for lightning to strike, I call these four-faced Buddhas. For lightning to strike 4 times in one person is not a sustainable model. You know, I doubt that the chances that any of us here have those 4 sets of skills, very, very low. So instead, what I decided to do was to try and create the Open Genomics Visualization Initiative, where we, instead of getting one person to do everything, we create consortia and have classical representational artists work with data visualization experts who are often found in the journalism world, depicting information for the public in a way that is understandable. The people who use the data, the physicians, the molecular biologists, the genomicists, the statisticians, and then computer programmers are the product makers today, the people who turn it into something that we can see. So I started working with Daniel Cohn. Daniel is an artist who was the founding artist in residence at the Broad Institute in Cambridge in Massachusetts. Daniel is an extraordinarily bright guy whose work greets you as you get off each elevator in the Broad Institute. It's the top genomics institute in the United States. And Daniel's work is, I find it striking, I find it really beautiful, and it's large. It's you know, half the size of this wall in blue here. It's a really imposing spectacle. So it was very interesting working with Daniel. He was challenging a lot of our ideas about what we should be thinking of in terms of how we represent data. But in order to be able to communicate with Daniel, I thought, I've got to meet him halfway. So we published a paper last year about epigenetic regulatory events in the human genome, where we're looking for deviations from the typical Watson and Crick double helix. There are other things that can exist in our genome. And we depicted a type of information in them using this red and blue plot. And this communicated 2 things to me. One was, it told me a lot about these structures and why there was something interesting about them from a scientific point of view. But the other thing was, it kind of got me there. I thought, this actually looks really pretty. It looks beautiful. So I did the logical thing for a physician scientist. I decided that what I would do is paint it. So I got a big piece of canvas about 2 meters tall, printed the image onto it, and then painted over it with oil paints. And I don't know why I was particularly drawn to that, except that it taught me that artists are seriously talented people. They're very patient, they're very good with their hand-eye coordination, but even still, with my sort of very crude attempts, the other artists in this shared studio would come and they'd say, what is that? Look, that looks really interesting. They could tell there was a story behind it. And so the idea that you're trying to communicate a story in art was something else that I hadn't quite appreciated. That's the original image. That's what happens when you start painting over it with oils. You lose accuracy, but you gain something else. There's light catching this because the paint piles up and you can see a certain texture to it. And there's something, you've enhanced it in a certain way while you've actually destroyed the accuracy of the information that it contains. So I was beginning to get Daniel at this point. I then started working with the second quadrant within this initiative, data visualization experts at the New York Genome Center and Nicola Stransky up in Cambridge in Massachusetts. So Nicola is famous within our community for having developed what's called the co-mutation plot for cancer genomics information, although everyone just calls it the Nicogram. And Nico, what he's done here is he's managed to put into 2-dimensional space an awful lot of information about patients with this particular cancer. You've got the genes here, P53, and a tumor suppressor here, and you work your way down. The DNA sequence change here, the patients are in columns, and the protein sequence change is depicted by these blue and green patterns. And he creates a heat map in the middle that allows you to see all of this stuff relative to each other. So you can see whether they're ordering themselves into any sort of clusters. Very interesting stuff. To me, Nico is a 4-faced Buddha. He's one of these guys who's able to do all of this. He programmed this himself. But what Nico has done is he's another French guy. He's created another Lua scaleogram. He's created an updated version of a heat map. So our question was, how can we out-Nico Nico? So we thought, right, why do we do things in 2 dimensions? We do things in 2 dimensions because the expectation is that we should be trying to get a paper in the New England Journal or in Nature or Science or something like that. But nowadays, very few of us actually read a paper version of anything, with apologies to Moorish. We are now looking at everything through digital media. We're using screens. So if we're using screens, we now have the ability to look into more dimensions than just 2. So we don't have to be as ingenious as Nico. So we created this thing with Nathan Pearson, which we call Picasso. And what we're looking at here is life expectancy, years, population size, age, and also painting on GDP, and painting on the different continents. And what you're able to do by having just two-dimensional representations, but moving from one to the other, is you start seeing the structure within the data, and you start seeing where individual continents, like Africa, has much lower life expectancy than Europe, for example. And you're able to see how individual data points move, how things cluster. To us, this is a nice prototype of where we need to be going in the future. We're working on having it so that you can zoom into it with immersive reality, where you can put goggles on and use your hands, but that'll be another time. So this was my epiphany. I went to IBM to talk about data visualization, and I put up this picture, and I said, Daniel is creating these beautiful images, but he's not trying to be accurate. And I said that, and the whole audience started creasing themselves laughing. And I thought, that's gratifying, but that was not meant to be a joke. And as I was coming home from this, I was puzzling out why they thought this was funny. And what I realized was that they're a bunch of engineers at IBM. To them, accuracy is the highest priority. And anything where I say that somebody's being inaccurate, it's a dig. They thought I was having a dig at Daniel. So it made me start thinking, why is it that Daniel is being intentionally inaccurate? And why I was drawn to this idea that I should take accurate data and damage it by creating this inaccuracy. Why did we both end up drawing to the idea of creating ambiguity? So the ultimate representation of data in medicine is a report of some form. You've either said, this is the diagnosis, or the radiologist has given you a report, or the pathologist has given you a lab report. But the penultimate representation of the data allows ambiguity and allows interpretation. If all we have to work with is this, we're robots. If we're working with this, we can apply our intuition. It's the same in science. This is an epigenetic modification of a gene that predicts whether kids with osteosarcoma will have very good survival or will die very quickly. It's one of the many loci that's being shown on this heat map. I give people this, I'd better be right. I give people this, there's room for other interpretation, perhaps improvement on the observation that we made. So the penultimate representation that allows ambiguity is very important. And this was my epiphany. What an artist is trying to do is not to create this single winning model, the single interpretation of his or her work. What he or she is trying to do is to allow each of us to have our own individual interpretation of the data so that we can form our own conclusions. So everyone who gets off the elevator at the Broad Institute is seeing something different. In medicine, we start with a differential diagnosis. We apply some sort of hypothesis testing to come up with the diagnosis. It's here that we could make a mistake, and it's here that we have the opportunity to educate, and it's here that we need to be clear about what we're trying to do. And a lot of the time that's buried in terms of the decision-making process and what would be seen in a chart. It's the same, sorry, let's go back for one more second. It's the same in science in that we have competing hypotheses. We do something to try to come up with a single hypothesis at the end, and we present this, say, in a paper or in a presentation like this. But if I'm wrong in what I've decided here, this is where you want to go back to try to understand what the alternative correct solution was. And to make this clear, we need to have some sort of ambiguity in the visual representation of these data that allow people to explore. It's critically important for reproducibility in research, which is a massive issue these days. It's a critical issue in terms of how we come up with our patient care. But it's also
something that's going to serve this generation's Barbara McClintock. And so to allow her to see the ambiguity in what we're doing in the course of our decision making, we need to have visualization. We need to get a bunch of people together to make this happen. We probably need to start exploring how to visualize in multiple dimensions, and we need to celebrate ambiguity in the same way as our artist colleagues do. And if we do that, we're going to go from what is currently preventing today's Barbara McClintock from making intuitive insights and allow her to get back to having a feeling for the organism and to have her own epiphanies. Thank you.